then large pre-B cells, then small pre-B cells, and finally mature B cells. As the cell develops, it makes permanent changes in its DNA, so that by the time it's an immature B cell, it has DNA that uniquely codes for a B cell receptor that can bind to foreign antigens but isn't self-reactive. The B cell receptor has two chains, a heavy and a light chain. The heavy chain contains regions that determine the type of antibody it'll be, as well as whether the B cell receptor will be surface bound or secreted, like an antibody. The region where the heavy chain and light chain together form a unique protein structure capable of binding proteins, carbohydrates, or lipids that the B cell might eventually encounter. And this is called the antigen binding site. The antigen binding site of the B cell receptor is made up of 3 protein segments that are called V for variable, D for diversity, and J for joining. The heavy chain is made up of 1 V segment, 1 D segment, and 1 J segment, while the light chain only contains a V and a J segment. And this is pretty easy to remember. Just think of it as the extra segment making the heavy chain heavier. Every person inherits multiple genes that encode the V, D, and J protein segments, and these segments can be mixed and matched to make a unique structure. This is kind of like how you might have several pairs of shoes, pants, and shirts, and you can mix and match them to create different outfit combinations. For the heavy chain, each person has 44 V gene segments, 27 D segments, and 6 J segments. And there are even more V and J segments for the light chain. So one B cell might have a B cell receptor with a heavy chain that has VH1, DH3, and JH5 combination, and a light chain that's a VL7, JL2. And then there might be another B cell that has a B cell receptor with a heavy chain that has a VH44, DH10, JH1 combination, and a light chain that's VL2, JL3. And that would mean that these two B cells